Please give your responses if I'm audible. So students, yesterday in my lecture, I talked about the GenBank file format, and as I shown you that since GenBank is not in detail in your syllabus, but DDBJ is in your syllabus. So regarding the background information of the DDBJ, I have already discussed with you, and the file format, the way by which the databases are showing their formats, the information. Just simply, you don't be scared about the format, what format is all about. Let me give you a very simple example, right? Suppose suppose just imagine that is the offline class in your classroom. Anil sir is taking your lecture, right? So Anil sir is taking lecture for common for everybody, right? So he's not especially teaching to any single person, right? So suppose 50 students are there in your class. So the content which sir is delivering to you, that is same for all. But at the end of the lecture, if teacher asks you that you show your notebook, right? Show your notebook, what you have noted down during the lecture. What do you think? Whatever you have written, that will match with your friend? Obviously no, not at all. Why it is not going to match? Because maybe some points you think it is very important, or you and your friends think that it is not important at all. So I will not write it, right? So for each and everybody, the notes will be different. Someone's writing will be very good, someone's writing will be very bad, someone will the moderate level. So the way you write the notes, that is your format, how you are making your notes. Likewise, since the databases are autonomous in nature, all the databases have their own administration. So very simple, the information, the way in which they keep the information regarding any biological sequences, that is going to be different. So that is simply known as database file format. So what is the criteria, what is the way how database are arranged the information, they are being putting the information in the database, that is known as file format. So I discussed in detail about the GenBank file format. So if in examination it comes, write a note on the DDBJ. So what you have to do, you have to just give the basic information about the DDBJ, what DDBJ is all about, who is maintaining it, what is the retrieval system, what is the submission system, who can access it, what type of information is being hosted in the DDBJ, and how DDBJ represents its sequence, that is known as DDBJ file format, and that is 100% copy pasted from the GenBank. So buy one get one free, right? If you know GenBank file format, means start with the locus, definition, SSM, the header, feature, and sequence itself. Whatever the things I explained you, that you also have to write down in the DDBJ file format. So if in examination, suppose the maximum marks is 7, right? So if examiner asks that write down the notes on the DDBJ, so obviously you have to give the background information, some technical information, and then you go for the DDBJ file format. And if they ask about the write notes on the DDBJ, then no need to go for the DDBJ file format. If you write the basic information about the DDBJ, where you can found it, what is the summation tool, what is the retrieval tool, who is maintaining it, since how many years, right? So all these basic details, if you give it, that will be enough. So now we are going to talk about the next format, that is known as EMBL format, and I told you now the name of the EMBL have been changed. The new name is ENA, European Nucleotide Archive. The basic about the ENA I have already discussed in the previous lectures. Right? The ENA is also the part of the INSDC, International Nucleotide Sequence Database Collaboration. Alright. And the submission tool for the ENA, can someone message in the group what was the submission tool for the ENA or EVI? What was the submission tool and retrieval tool? Anyone can answer? I am waiting for your responses. What was the name of the submission tool and retrieval tool for ENA? And since ENA don't have any separate website, to access that you have to go for the composite database, that was the EVI. So simply my question is, what was the name of the submission tool and retrieval tool of ENA? Can someone do respond me so that I can come to know that you papers are very sincere and you are noting down the things? The straightforward questions I am asking to you. I am waiting for your answers. Once you answer me, then I will proceed ahead. So maybe your comments appear to me later. So let me continue the session, and we'll check later on who have given the answer. So the EMBL file format, the basics I have already told you about the ENA, and just like I told you, if you know the GenBank file format, the same file format will be adopted by the DDBJ. Likewise, if you know the file format of the EMBL, which is now known as ENA, the same file format, one another database is there. The name of that one is the SwissProt. SwissProt, I think you know the SwissProt, although it is not in your syllabus, so you don't have to bother for anything. So the same, whatever ENA file format having, the same way there is the SwissProt file format. So you see now question, someone may arise the question. So sir, if in the examination it comes that write down the file format, then what should I write? Should I write the starting from ID till the double slash sign? Answer is no. What you have to write, whatever the things are in the right side, right? Like here it is ID, right side, whatever the things is there, that you will not be able to remember any time, and it is not necessary also. The only thing you have to discuss in the examination, what is ID, what is AC, what does it mean the DE, what does it mean the FT, the SQ. These are all the things, the abbreviated forms, that only you have to write in the examination. Whatever the things in the right column, that you don't have to write, and obviously it is not possible for anybody. Say for example 25 crore sequences are there in the database, so obviously the whatever information is being in the right side column, that will be different for all the 25 crore sequences. So it will not be able, you will not be able to mug up all the things, right? So it is not necessary. You have to just only write down what ID means, what is DX means, what is double X means, what is AC means, what is DE means, what is FT means, what is SQ means. That only you have to explain. Okay, so another example, it was very simple, very small example. So I am showing you one more example. Say the SSN number is AA03518. So this is the bit bigger, so that maximum number of keywords you can mug up. See here the ID. If you compare this file format with the GenBank, if you remember, the starting of the GenBank file format was started with the locus, right? But here in the EMBL file format, since I told you the databases are autonomous in nature, so each databases have their own format, right? If I am making the notes for myself, you are making your notes. So the information is almost same, but the criteria, the way they represent the sequence in the database, that is bit different, right? So just like in the GenBank, the starting of the file format was with locus. Here, the EMBL file format was starting with the ID. ID means what? Identification number or identifier. You can say identifier. So under the identifier, there is many things like SSN number, right? Then SV means sequence version, which is 1, means whatever the sequence was submitted first time. Till then there is no modification, just like I told you the first edition of the book. Then what is the topology of the molecule? That is linear. The same thing we discussed in the GenBank. Then it is genomic DNA. Then the division, it is standard division. Prokaryote, the sequences are from prokaryote. And how many base pair it is? 1468. So you will notice here that almost everything is same except the order, right? Except the only orders different, otherwise maximum things are same, just like in the GenBank. Then double X. This double X is known as spacer, that don't have any meaning at all. Why we are putting why database is putting the double X? So that you can read it in a human readable form. There should be a gap in between the sequences, right? So double X don't have any meaning. It is just simply spacer. Alright. I think there should not be any question. If you have any kind of the question, you please ask me. I will very happy to answer you. Okay. So till I was just checking your messages, no one gave the answer. So let me give you the answer of the ENA. The submission tool for the ENA was known as Webin, if you remember, and the retrieval tool was SRS, Sequence Retrieval System. Please note it down. The name of the submission tool was Webin, and the name of the retrieval tool was SRS, Sequence Retrieval System. Right? Now, the next line after ID, it is AC again. It is repeating. AC stands for, please note it down the full form. AC stands for SSN number, which is SQ190916, the same sequence which I explained now in GenBank file format. The same thing I am explaining in the form of the ENA, EMBL file format. The DT stands for date, 7 September 2010. And it is just showing DT is repeating twice. It may repeat thrice, 4 times, 5 times, there is no limit. So suppose 5 time updation was there. So there will be the 5 time DT repeat. If it is not a single time repetition, a modification was done, so there will be 2 time only. So 7 September 2010 that was released, and last updated was 7 September 2010, means it is version 1, means only one time the sequence was submitted by the authors. Again the spacer, DE, please note it down. The full form of the DE is known as definition. Definition, what do you say? Definition. So in the definition it is showing you Selenococcus roseus strain RVSC OKH1 16S ribosomal RNA gene, and it is partial sequence. So since the definitions is carry forward in the next line, so D is repeating twice. If the definition is large, then D may repeating 4 to 5 times or more than that, right? So D doesn't guarantee that it will come only once. If the definition is continued, D will continue. Then spacer. Keyword, if you remember in GenBank that was also left, the authors have left it blank, so it is blank. Again XX means it is spacer. Organism species. OS, please note it down. OS stands for organism species. Then OC, organism classification. So here you find the name of the organism here under the OS, and the detailed classification of it you will find under the OC, this is organism classification. Again spacer, a reference number, just a minute. Yeah, so sorry for the bit interruption. So students, I was talking about just like if you remember why I am giving the same example, so that you can compare, right, from the GenBank file format and from the EMBL file format. So in that, if you remember, in the GenBank file format, the references I told you, suppose the author, by using this particular sequence, whatever you are watching right now, if by analyzing that, researchers have published 5 papers. So they're starting from the RN references. I told you that 5 time repetition will be there, right? So here it is in bit abbreviated form. It is RN means reference number 1. RP, please note it down, reference position. Reference position means what? Reference position means, suppose the length of the sequence is 10,000, right? So on 10,000, suppose from 1 to 2000, by analyzing, you have published one paper. and analyzing 2001 to 4000 you have published another paper, right? So that you can put the reference position here, since the length of the sequence is only 1468. So here it is reference position is 1468, 1 to 1468. RA, please note it down the abbreviations. RA stands for reference author. So these are the author who have done the sequencing. You can find my name also. So my sequence is available in the GenBank. RT means reference title. Reference title means if suppose I publish a paper, then obviously there should be some name of the paper, right? So that we may find under the reference title. We will find under the reference title. RL, reference location. Please note it down. Whatever full form I am telling you, please note it down. RL stands for reference location. Reference location means what? In which particular journal you have published the paper. So that will come under the RL. So since the researcher have not published the paper, so it is directly written that submitted to the IRSDC and Sri Aman in Virani Science College, Rajkot, Yogidham, Kalaudu, Rajpur, Gujarat, something. So RL means reference locations. If they publish the paper, then you might here you might see the name of the journal. Since the practical portions I have already shared with you, so by following that you can verify it, and you also can retrieve other sequences from the concerned databases. DR. If you remember, while I was talking about the features of the biological databases or characteristics of the biological databases, no, in that case I told you that the feature of one database is known as integration with the other databases. Integration with the other databases means what? Here it is showing you DR. DR means database cross reference. So the similar type of information is also available in another databases, right? So all the R suppose whatever family, whatever RNA sequence it is genomic DNA. So the same is available with the RNA family. So these are the different links available. If you want to explore this particular sequence in detail, then you also may visit these are the different databases. Then FH, feature header. Feature header I told you, in the GenBank there was three things: the header, feature and sequence. So whatever the common things will be there, that will be under the header. Then the feature. So header, under the feature header it is written as key, location, qualifier. So source, you can see the feature table, all the annotation regarding this particular sequence you may find on the FT. FT is continued, and finally you will see the sequence. Just like in the GenBank, in one row there will be 60 bases, and it stops with the greater than sign. I mean double slash. So almost the things are same in the GenBank file format, in the ENA file format also. So what I am doing now, just today I am going to share all the PPTs whatever I taught you till dated. So you do one thing, you please go through that. Regarding the DNA data bank of Japan, like here I have also given the notes. DNA data bank of Japan, whatever I explained, that each and everything is written here. So whatever I have till dated I have taught you, that I am going to share with you. You please go through it, and meanwhile if you are facing any kind of the problem, you please may ask the question. Alright, so that's all for the databases like DDBJ and ENA. Alright, now I am going to teach you the another database. The name of the database is known as PDB. That is in your syllabus. Another database. PDB stands for Protein Data Bank. What Protein Data Bank means? As name suggests, Protein Data Bank means what? In the Protein Data Bank, PDB, you will be able to get the 3D structure of the proteins. So let me open the 3D structure of the proteins. So it is rcsb.org. If you want to retrieve the 3D structure of any protein, although I am telling you protein, but it is not true actually. You may find the 3D structure of any macromolecule. Macromolecule, what comes under the macromolecule? Under the macromolecule, it may be the protein. It may be the lipid, it may be the protein RNA complex, it may be protein DNA complex, it may be protein carbohydrate complex, it may be nucleotide carbohydrate complex. So whatever the 3D structure of any micromolecule is available, if that is solved by any researcher, suppose you are the researcher and you identified a particular protein, you isolated particular protein from any particular virus, then with the help of there is three method, okay, how you can determine the 3D structure of any particular protein. The 3D structure, I think you have shown, if once you know gone through any biochemistry book, you will find the beautiful pictures of the proteins. The here you also see here one protein. You can see the protein here, the structure, the 3D structure. How beautiful it is. It is made up of beta sheet, the alpha helix, then loop, and all, right. So all these 3D structures you may find. So what I was telling you, suppose you isolated a protein, and with the help of three methods are available. That is, one is X-ray crystallography, another is electron microscopy, and third is nuclear magnetic resonance, means NMR. So there is three methods available: X-ray crystallography, NMR, and electron microscopy. So by using any of them, scientists are doing the 3D structure determination. Alright. So suppose you are skilled with the X-ray crystallography, and with the help of the X-ray crystallography, you have solved the structure. So obviously, whatever 3D structure you got for any particular protein, that you are not going to keep with you. What you will do? You will submit to the PDB so that other researchers who are working on the same field, they can access the data and they can visualize, they can analyze that, right? So just like I told you in the GenBank, if you want to submit any kind of the sequence, you need one tool. That name of the tool I told you, like for GenBank, it was Bankit, right? And if you want to retrieve the information from the database, that was the Entrage. So likewise, if you want to, if you want to submit your structure, 3D structure of protein, here the option you may find deposit, right? Option, the simple option you are getting, like deposit. Here you get a lot of options like prepare data, validate data, deposit data, right? A lot of options are there. Definitely you people are not from the crystallography field, so you will not able to understand. So data, some here is there, some other options are available, like how you can prepare your file for the deposit. So here is a Maxit, translate data between file format and more. So by using these methods, you will be able to submit your sequence to the PDB, and once that will be validated by the database administrator, that will be publicly available for the access, right? So if you want to search any particular protein sequence, you also may do that. If you remember, the two videos I shared, in one video I talked about how to retrieve the protein sequence. In another one I told you how to get the protein sequence from the Swiss prot, if you remember. Likewise, if you want to search any of the proteins, then you can search from here also. So just like in the GenBank file format, GenBank, there is the SSN number. Likewise, once you will submit your structure, in return, database will give you one PDB ID. PDB ID is of 4 characters. Alphanumeric means number will be there, as well as alphabet will be there. So it is just like the SSN number. So if you have the SSN number, you can put that SSN number in this particular field, and the 3D structure of that particular protein, you will be able to fetch it out. So before I proceed to demonstrate how to search for any particular protein, let me give you some more statistics, right? Like how many structures are being deposited right now in the PDB. So here you find currently 1,77,910 biological macromolecular structures are there. 1.77 lakhs structures are there. as its name suggests, it is protein data bank, but it doesn't mean that only protein sequences are there. You will also find other macromolecules structure. If you want to see, here you see, I am telling you here and that the analyze one option is there, PDB statistics. If you click on the PDB statistics, see here is the statistics given for the PDB. So what are the PDB? See here, what are the statistics you see here? See, as I already told you, there is three methods for the protein structure determination. So if you want to know that how many structure have been solved by X-ray, so if you click on the by X-ray, out of 1.77 lakh structure, you will be able to know that how many structures have been solved by X-ray method. So you can see 1.6 lakh, around out of 1.77 lakh structure, 1.6 lakh structure is solved by X-ray method only. So X-ray crystallography is very popular method and most recommended method for the 3D structure of the macromolecules, right? And if you go for you want to know how many structure have been submitted by NMR, so if you click on NMR, out of 1.77 lakh structures, you just come to know that only more than 13,000, around 13,000 structure is solved by NMR. So this is the second method, and the least recommended method is electron microscopy. If you click on the electron microscopy, I think you are able to watch the things very clearly. Here you will find that less than 8,000 structures are solved by electron microscopy. So means you are here, now you are able to conclude that the most recommended method for protein structure determination or macromolecular structure determination is what, bachcho? X-ray crystallography. And if you want to know that how many in the protein data bank, is there only protein structures or other structures are also there? The total structure, you know, it is 1.77 lakh. So if I click on protein only, see here you will, again you come to know that approximate 1.6 lakh structure is of protein and remaining is how many? Only 17 lakhs, because 1.77 lakh structure was there, no, total. So almost it is approximate 1.6 lakh structure is of protein and remaining in 17,000 there is the lipid, there is the carbohydrate, there is the protein and other combinations, right? So major part is a protein. That's why this is known as protein data bank. It is specialized in protein structure, that's why. Alright. So now I am just going on the home page of the PDB. Let me teach you, let me demonstrate. If you want to retrieve the structure of any particular protein, how you will be able to search. So two things is there. The number one, either you should have the PDB or you should have the keyword. So obviously I don't have PDB, I don't have right now. Suppose I don't have the, so you put it any of the protein which you remember if you want to see the 3D structure of that particular protein. So simply very crude type of a query I am doing. Suppose protease. Here is spike protein. If I do spike protein of coronavirus, right? So if I do spike protein, let's check it out. What is the spike protein of coronavirus? See here, it is automatically suggesting you. Once I put it the spike SARS-CoV-2 viral proteins. So let me click over here and just check whether the 3D structure of the spike protein of coronavirus have been solved by any researcher till dated or not. So it is not giving the result. It depends on the internet speed. If internet speed is good, you may get the result in a fraction of minutes, otherwise you have to wait for some time. Yeah, see here. It is showing you 7 BNM. I told you, no, that for each and every protein there is the one PDB ID. So the PDB ID is 7 BNM. It is the closed conformation of something spike protein of SARS-CoV-2. So if you want to see the 3D view of this particular protein, what you can do here, it is showing you 3D view. So if you want to see in 3D, see here, just click on the 3D view and just wait. This protein, this spike protein of the coronavirus is going to be shown in the 3D format. Just wait. It will download all the applets and everything. Technicalities are there. Since the file is larger, so you have to wait for some time. Depends on the internet speed. Just wait for a few seconds. Yeah, so dear students, now you can see the beautiful structure of protein, right? And if you want to zoom it, just simply what you do in your mouse, right? This thing you will be able to do more accurately while you are using the laptop or desktop. So in your mouse, the circle that is there, no, the wheel is there, no. So wheel you can rotate. So once you will rotate wheel, the structure will be zoomed in or zoomed out. So here very clearly you can see the 3D structure, the beautiful structure of a particular protein. Another, by using the wheel you can zoom in or zoom out. If another things, if want to, suppose I want to locate where is the M. See here the amino acids, right? If I click, once I click on the M, so it is going to be highlighted. Any of the amino acid. Once you click on any of the amino acid, that will be highlighted. Alright. So now if I want to come back, just click on this circle, the reference. Suppose I want to locate the valine, okay. So if I click on the valine, where valine is all about, so you see here, the valine is zoomed only. Now if I want to reach in the original position, just simply click on the refresh, you will find the original structure of the protein. And now what are the different way you can, just if you right click, I think some other options will be there. There is separate softwares available like PyMol, JMol, MolMol. Different softwares are there. By using that, you will be able to uh, analyze this particular protein in more interactive way. So here, just what I want to show you, that how to analyze how to see the 3D structure of any particular protein, and how you will be able to analyze it. If you left click the mouse, you can rotate the protein also, rotate in 360 view. Although there is very less functionality is there in the PDB. So if some scientists want to analyze this particular protein in more detail, then what option they have? Suppose I am the bioinformatician, right, and I want to analyze this particular protein structure in more detail. So what I can do, just simply I can download this particular file in my computer. Click on just simply download file. See here, automatically 7bnmcif.gz. So this protein structure have been downloaded in my computer, and if I use another softwares and I am expert bioinformatician, then I will be able to analyze these protein structures in very detail. So this is all about the PDB. That is not in your syllabus, and it is not necessary also. That's what I'm not going to teach you the analysis of the proteins, right? So the only things you have to keep remember, what is PDB all about. Let me summarize in very quickly. Let me summarize what is PDB. PDB is the full form of the PDB is the protein data bank, right? And currently more than 1.77 lakhs of 3D structure of proteins, carbohydrate, lipid and all available with the PDB. There is three methods for the protein structure determination: the X-ray, the NMR, the electron microscopy, X-ray is the most recommended method, right? Then and maximum the number of structure is available in the PDB is the what protein. That's why it is written as PDB, protein data bank. All right. So that the basic introduction, and you can download, you can search any of the protein structure. If you know the PDB ID, it's very simple. You can put it here, and the only one structure you will find. Or else, if you don't have the PDB ID, in that case, you can type the name of the protein and related structures you will be able to get it. Since you are getting three structures here in this case, now you have to take the decision in which particular structure is of your use and in which you are interested to analyze, right? So this is all about the protein structure, protein data bank. Tomorrow we'll talk about some enzyme database and we'll talk about some other databases. So tomorrow again I will see you at sharp 1 o'clock. Till then, the videos are available in the playlist in my channel. I hope you each of you have subscribed the channel. Date wise recording is available. If any of the lecture you miss or you forget to make the notes, you please replay those structures and you can make your own notes. So see you tomorrow with the new topic at 1 p.m. Till then, take care. Have a nice day.